Hi everyone, I'm Dennis Parker from the University of Utah. I would like to thank the organizers of the meeting for this invitation to participate. I do have some disclosures on IP related to MRI-guided focused ultrasound. I would like to talk about two challenges still facing focused ultrasound brain treatments. First, there is still work to be done to improve the consistency of ultrasound beam focusing through the wide range of skull anatomic variations. Second, there is work to be done to improve monitoring of the heating around the focal point as well as throughout the skull and cranium. The need for focusing is illustrated nicely by these images from John Snell, showing the refraction effects from the variations in skull shape around the skull. You can see there. The distortion of ultrasound propagation through inhomogeneous media can be monitored using full-wave angular spectrum numerical modeling, but these techniques are very slow. Doug Christensen and students at the University of Utah have developed a very rapid full-wave approximation method called hybrid angular spectrum that uses fast Fourier transforms to achieve up to 2 orders of magnitude increase in speed. The Stanford focused ultrasound group led by Kim Beltz-Polly implemented HAZ to model beam focusing and heating achieved in clinical essential tremor treatments. This is showing some of the results from the paper that they published as referenced there. We are also in the process of modeling essential tremor treatments, transforming the CT volume images into the treatment orientation. We construct a model of tissue types, including brain, bone, and diploid, as shown here on the lower right. Then using the specified treatment focal positions and tissue acoustic properties, we apply HAZ with time reversal to determine the transducer element phases to achieve focusing at that point. We then use HAZ forward modeling to predict the beam intensity of energy deposition pattern for the time reversal phases, as well as that for no phase correction and for the phase correction used in actual treatments. This is shown here, where after we get these heating patterns, we then apply the Penny's bioheat equation with published tissue thermal properties to obtain patterns of temperature rise. The differences in heating patterns can be seen comparing no phase correction to the clinically used as well as time reversal for this specific model using HAZ applied. The simulated temperatures can then be compared with measured MR temperatures. Current results of these simulations are given in a poster presentation here by Emma Slominski, one of our students. A second use of this will be to use time reversal phases to improve beam focusing and actually test the accuracy of acoustic models, and that's work in progress right now. I'd like to briefly mention the challenge of monitoring temperature with MRI, and this can be monitored using the proton resonance frequency method, as seen here. However, a single slice 2D PRF method cannot see heating throughout the skull and brain, and a full 3D volume measurement really is needed. This can be seen, let's see if I can get this to run, in these actual heating applied to a gelatin-filled skull using the Insightec system. Heating at the nearby bone surfaces can be equal or exceed heating at the focus and should be monitored. And we and others are working on 3D, fully 3D MR temperature imaging methods for these studies. Finally, I would just like to mention the banding artifact in brain treatments that causes a reduced measurement accuracy at the focal position. Rock Hadley at the University of Utah, Will Grissom at Vanderbilt, and others have studied methods to move this artifact. Rock has used a conductive screen that is highly ultrasound transparent to move this artifact away from the focal position. And you can see that here, and the screen is in two positions. One where it's slipped off to the side and actually moved the banding artifact quite a bit away from the focus. Another where it's in a more expected position and moved the banding artifact quite a ways away from the focal position. Finally, I'd just like to mention, because of the unavailable of our MR-guided focused ultrasound system, except very late at night and on weekends, we have designed and constructed a mock system, which actually represents the clinical system quite nicely. And this will allow us to do our experiments much more efficiently. We're looking forward to using this. This is hot off the press. So with that, I'd like to acknowledge our group. We have many researchers and clinical collaborators, students, and staff involved with us, and I appreciate you allowing me to present here at the meeting, and thank you for your attention.
Hello, my name is Craig Meyer. I would like to talk about some recent developments in MRI guidance of brain fuzz. First, I'd like to talk about hotspot thermometry. This is some work by Will Grissom. The idea here is to use a two-dimensional RF pulse to both select the slice and to suppress the signal from the water bath. You can see in the center image that the water bath signal is largely suppressed on both the right and left sides of the image. This results in a nice temperature map, and it has two advantages. First, it leads to higher precision temperature maps. Second, it allows the scan time to be cut by a factor of about 2. Next is some more work from Will Grissom's lab. In this case, it's simultaneous multi-slice thermometry. The idea here is that you simultaneously excite 3 different slices, and then you sort of disentangle the slices in the image reconstruction. The center image shows temperature precision maps, and you can see that the precision remains high even when you're simultaneously getting 3 slices in the time it would normally take to get 1 slice. Volumetric coverage is important to avoid missing the location of the peak temperature and to improve efficiency. My lab works on 3-dimensional spiral thermometry. Spiral imaging is a way to scan rapidly using MRI. We've gotten some promising results with this method, as you can see in the center image. On the right, you can see that the 3D thermometry accurately predicts the ultimate lesion size as defined by T2 weighted imaging. While this method is fast, it can be accelerated even further using the Kalman filter. The idea here is that we subsample case space, and then we fill in the missing data using a Kalman filter, which is a type of learning filter that does not require pre-training. This works much better than a standard method known as view sharing. Next, I'd like to move on to skull heating. We currently do not monitor skull heating using MRI, but we believe that it might be possible. Once again, we're using a fast 3D spiral pulse sequence. This one is different in that it has an ultra-short echo time, which means that the bone, which normally doesn't appear in an MRI scan, appears bright in the image. Also, we're using T1 mapping instead of T1 weighted imaging, because our results show that it will be a more reproducible way to visualize regions of skull heating. Katja Gilbo is going to present this work at the symposium. Here's our experimental setup on the left for validation in cow bones. And here are some of our results on the right. There are good linear fits between T1 and the change in temperature. We believe that this method has a chance to detect skull heating in patients. Next, I'd like to move on to treatment monitoring. The goal here is to provide guidance to avoid undertreatment or overtreatment. Diffusion may be an early marker of ablation, much as it is an early marker of stroke. diffusion imaging in the transducer is a hard problem. And Stephen Allen has developed the spiral pulse sequence on the left, as well as a motion compensated image reconstruction method. And this enables in vivo diffusion imaging. Stephen will present this work at the symposium. You can see on the right that the T2 weighted image doesn't show the lesion 5 minutes post-ablation, whereas the diffusion weighted image shows it well. Here's a statistical analysis of 25 ablations in 6 pigs, and it confirms that diffusion shows up earlier than T2 weighted imaging, and that it provides a better marker of treatment completion. Lastly, I'd like to go back to the water bath. This is also work by Stephen Allen. The idea here is to dope the water bath by adding iron oxide nanoparticles, which suppresses the signal that comes from the water bath. You can see in the lower right that the water bath shows up brightly without the nanoparticles, but with the nanoparticles, the water bath signal is largely suppressed, thus reducing artifacts and allowing a smaller field of view. Stephen will present this work as well at the symposium. I look forward to the Q&A session with my colleagues at the symposium. Thank you.
Well, welcome to the Brain Technical Imaging panel. We have some distinguished guests today to discuss brain imaging. So this is largely going to be an MR-centric discussion. We have Dennis Parker and Craig Meyer and Will Grissom with us. Thank you, gentlemen, for being here. We all met, I think, in 2012 at a meeting we had in Charlottesville back when treatment for essential tremor with transcranial focus ultrasound was kind of a new thing, and there were some issues that we were struggling with then. I wonder whether you guys could comment on, what some of those issues were and maybe how they plotted a path to where we are today with MR imaging research and transcranial focus ultrasound. Dennis, go.
That requires memory. So, well, obviously the thing that we were most interested in was we were trying to measure temperature in 3 dimensions. We were doing it primarily for non-brain applications, but when we came and saw what was being done, we believed that it would be important to measure with some sort of a 3-dimensional technique, and that became a focus that we had for quite a while. The other major problem that you seem to have with an ultrasound system that large are the problems associated with the water signal, that it tends to move around and vibrate and cause problems that cause artifacts and just problems where you get noise due to the water. So those 2 main problems were the obvious ones to me, but then obviously there's a lot of other problems where you want to really see where you're going and get better signal to noise. And with not being able to put a coil inside, it's quite difficult. So those were the things I saw. I don't want to take away. Craig or Will can add to that.
Yeah, if I remember correctly, at the time, one of the big things that we were spending a lot of time discussing was about the chemical shift artifact that you get in the temperature maps. As you're heating, the hot spot keeps blurring itself further and further. And so one of the big ideas that we floated at that meeting was doing multi-echo thermometry instead of single echo with a really long readout window. I remember we all gathered at the scanner and just coded up just in a protocol a quick multi-echo scan, and this seems to do the trick. And then GE we got pestered GE until they released a new a multi-phase type sequence that would work with multi-echo, and I think that's now basically the standard thermometry method, right, for guiding thalamotomy.
And I think, we also had sort of a zoo of artifacts that we went through. I think some of them were overranging and so on. What's become with that?
Well, yeah,
overranging.
Yeah, go ahead.
Oh, sorry. Overranging in which sense? In the
I think, like, again, you guys are the experts, but there were some pre-scan issues that were causing some clipping and other things.
Yeah.
Yeah. The water bath is a problem for doing a standard pre-scan. And so that was one of the things that we worked on when we later got together at UVA, at the University of Virginia, to try some different methods. Is that the pre-scan was not reliable, especially when we were using sort of other ways to control the scanner. And we've worked that out. We figured out how to do those pre-scans, and so we can then get all of the different methods for volumetric thermometry and other imaging to work pretty well, from the different groups, these groups and others as well.
Mm-hmm. So Nathan McDonald has asked, would flowing water doped with iron cause phase artifacts in the brain?
Yeah, so this is probably mine. So Stephen Allen has been working a lot, and we have a group of people also at Virginia Tech that have been working on working on doping the water bath to get rid of the MRI signal. And yeah, I don't know. I don't think so. But often you turn off the flow of the water anyway during portions of the procedure. But I don't think that it's going to cause a lot of artifact because there's just not much signal from it. And I don't know if you're worried about, Nathan, I don't know if you're worried about whether that might affect the signal in the brain. And I just don't think that the kind of concentrations we're using, that it's going to sort of go very far into it's going to go past the skull of any artifacts, because basically all the signal is gone. So the question is whether or not you're going to have T2 star signal loss in the associated skin and skull. And I don't think that will extend very far in at the kind of particles we're using, but maybe. We'll see.
The main concern is really the cavitation, right? The potential to.
Yeah. We're definitely concerned about cavitation. I'm less concerned about phase artifacts in the brain. You're right that it will have some effect on the bulk magnetization, but usually the iron oxide particles have a fairly confined spatial effect on off resonance and phase artifacts. So yeah, we are definitely concerned about sonic. About cavitation in the water bath.
The other problem that we wanted to put manganese in, and we're told by the manufacturer to not put anything in because of the risk of damage to the oxidation and everything else. But I don't think if you technically could do it, if you can kill the signal from the water, then I think it's going to be a definite step forward. I think the motion of water that has no signal, for example, if you were to use heavy water instead of protons, I think the phase from the moving water would be much less of a problem. The fact that it's moving around a little bit might cause a little bit of phase, but I don't think if the volume changes or the shape of it changes, I don't think there would be any artifact. I don't know. It'd be interesting to.
Nathan was just clarifying he meant issues with the static field.
Yeah, we'll look at that next time we're doing experiments. We'll see if the water changes the phase background of the imaging. That's a good idea.
So let's get back to thermometry for a minute. I mean, for people who aren't aware, measuring temperature during the treatment is still clinically done just as a single 2D acquisition, meaning we don't know what's happening out of plane. So maybe each of you could just briefly say kind of where we are from your perspective with 3D thermometry, that is, measuring temperature in real time of a volume, at least a slab around the focal spot.
Yeah, well, I think that without a major advance in coil capabilities, it should be possible with EPI or spiral type techniques to get to at least a few slices around the focus using current with our current hardware capabilities. And those just kind of need to be implemented and proven out. The work kind of needs to be done to just implement those and prove them out, I think. If we want to get to whole brain thermometry, then it's probably we're going to be requiring potentially some new hardware capabilities, some new receive coil technology that I know various groups of ours are working on. yeah, that's my perspective right now. Maybe Craig and Dennis have a finer point on that.
No, I think that's exactly right. I think segmented EPI has been our workhorse just because it was easiest to implement. But and I was disappointed because the segmented EPI does not do as well in the way that we've implemented as we would like because of sampling duty cycle. And we've got ways that we're going to try to improve that, and it may fare a little better. We're also working on a radial technique and very interested in the spirals that Craig's been doing. I think there are ways to do this. Well, Craig and Will both have been doing clever techniques to use non-Cartesian methods. So I'm optimistic, but I think it's really important, even if it's just around the focal spot. There's some real advantages to 3D, which not the least of which is you can interpolate, and you could do it in a good way. Anyway, sorry.
Yeah, and so we've worked a lot on the spiral stuff. We're optimistic that one of these trajectories, whether it's EPI or spiral or something else, are going to be able to generate the data that we need, and then basically the idea is just to get the hotspot and get the volume, get it, good image quality, accurate temperature estimates. And some of this involves both Will and Dennis in our group. All of us have worked on ways to accelerate the imaging. In other words, to fill in missing data using various different types of models. And so the combination of fast ways to acquire the data and then a little bit of additional acceleration in their image reconstruction, yeah, it should work, but we need to prove it out clinically. And so we've got a lot of preclinical data for this, but we need to continue to refine the methods and then make sure that it works in clinical testing down the road. So, and that'll be working with manufacturers, of course.
So at the moment, of course, we're constrained to either using the body coil or as it becomes available, maybe a 2-channel receive-only coil that would be a little closer to the head. But it sort of sounds like we're limited in lots of ways by the coil that we have available clinically. Maybe you could discuss a little bit about, what your hopes and dreams would be for having some kind of head coil for this technology.
Yeah, we kind of come together on this and have gone back and forth, and we've actually generated a coil statement of need about exactly what we'd like to see and what impact we'd expect a new coil development to have on the field, and in particular having coil capabilities that are even within maybe 80% of the capabilities of a sort of flagship 32 channel sort of head imager at 3T now would enable you to do really should enable you to do better diffusion imaging or DTI even to assess connectivity as a in situ treatment response monitoring. And then it would further allow us to do whole brain thermometry, which of course would make off center sort of targeting safer. And so we've actually generated a sort of statement of need where we said we would love to see a coil that provides SNR within maybe 80% of a modern head array, enables scan acceleration factors by K-space undersampling, which aren't really possible right now unless you further leverage the temporal dimension of the thermometry. And then of course it couldn't interfere. Such a coil could not interfere with the fuss beam or with water circulation or with the frame, and those are all really hard challenges. And it should be possible to use it with or without the water bath filled with similar levels of performance. Those are the sort of 4 bullet points that we came up with when we circled around this question.
And there's some progress on the way. The manufacturer is going to has made available in other countries and will make available in the US, what's called a Princess Leia coil, a 2-channel coil that sits in the water bath, and that will help. And but yeah, to really do advanced imaging, you need multiple channels. And you also, as Will perhaps wants to talk about methods to fix the B1 problem, although I'm not the moderator. So in other words, it's not just the receiver coil, but it's also, what's the transmit look like?
Yeah,
so I think like Will and Dennis have maybe contributed to the solution of that.
Yeah, we kind of came up with a very similar idea at the same time, independently. The idea being that if you just place some kind of metallic structure above the head, and it can be pretty high gauge wire structures, then such that you can minimally interfere with the ultrasound propagation, then you can sort of control and tailor the reflection of the RF waves that come in from the bottom of the transducer in the way that they bounce back down to constructively or destructively interfere with the incoming waves in such a way that you can get a total transmit field that is more spatially uniform and stronger. On our end, we got an NIH grant to keep working on this, and recently we got our first in vivo results using just 2 wires placed in sort of a propeller beanie configuration above the head. And it's looking pretty promising and pretty robust positioning. Our initial in vivo results, we can't put a healthy volunteer in a frame, so they're just kind of free floating inside the transducer in the water. And even with the propeller beanie sort of sitting back and their head near the back of the transducer, it's a pretty huge improvement to the B1 fields as we measured with a B1 mapping sequence. But we have yet to prove it out. So yeah, and then Dennis is also working on this.
So yeah, Rock Hadley in our group. So our thought was to just come up with a coarse screen. I think it's not as elegant as Will's solution in the sense that he's using Yagi-Uda reflector, if you will. Ours is a screen which is coarse, and it seemed to, the first times we tried it, it had a huge impact on just totally pushing the artifact out. But it's the same principle. It's disrupting the standing wave.
So we're coming down to the end of our time. So I think I want to switch gears a little bit, and as we move forward, some of the newer indications that we're looking at in the brain are using non-thermal effects, and may and probably will ultimately be, these procedures will be done outside the bore of a magnet, but MR will still have a role. And, as we're talking about BBB opening and drug delivery and maybe even histotripsy and neuromodulation and these kinds of things, what's your vision of future research in MR as we kind of shift? I think we'll always be doing things in the bore, but there'll be these collection of things that we're doing outside the bore.
Go ahead.
Oh yeah, we're doing some things along these lines. For example, with our HEAL project, we're trying to use the MR mainly to guide where the ultrasound will go, but it's going to be stuck. The ultrasound transducer is ultimately going to be stuck on a rigid mounted frame. We're going to use the MR in RFI to figure out where the focus is. When a subject who's receiving some kind of perhaps a DBS placement is in the magnet, we're going to figure out where the beam is, and then take them outside and potentially tickle the brain outside the magnet later on, using the MR as ultimately connecting the two via optical tracking and the fact that we know that there's a rigid frame attached. So we're trying to make this link in various ways. It's a little hazy still how it's going to come out. For that particular application, what we're looking at there is treatment of neuropathic pain, and we don't want to have to put the subject in the scanner every time that we're doing it. Just maybe do 1 or 2 procedures in the scanner, and then take it out. So we're starting to build these links of how you can establish your coordinate system in the scanner, and then be able to do accurate targeting outside the scanner using different optical tracking and frame type concepts. Mm-hmm.
Craig, what do you think?
Yeah, sure. And this is something of interest in non-focus ultrasound areas as well. Is the image fusion and image guidance and the synthetic MRI, or there's some work on doing synthetic MRI by pre-acquiring MRI and then doing it outside the scanner. But yeah, we're generally just image fusion for images that are acquired before and then after. We're already doing that with CT, and that's certainly going to be possible. And there's lots of interesting work in that area. So yeah, that would be a good thing. Also, the coil. If we get the coil and the B1 fixed, a lot of these things will be able to be done within the scanner at at procedural time. But yeah, initially things like, a lot of more advanced imaging needs to be done outside the water bath.
Quickly, Dennis, do you think there's a role for functional MRI and transcranial focus ultrasound?
Sure. Of course there is, and DTI and tractography, and but no, certainly the problem right now to get functional MRI working in the system is the challenge of signal to noise ratio, where you're not having a really, really good coil. So
yeah,
I mean, the bottom line is yes.
We're back to a coil. We're back to a coil, right? So
yeah, well, and that's why I wrote in the chat that there's a good chance that smaller transducers for blood-brain barrier opening and lower water volumes and just things that are consistent with building a good coil around it are coming.
Okay. Well, I think, gentlemen, we're going to have to leave it there, but I thank you so much for participating on this panel. Thank you to all you in the audience for your attention.
Thanks for having us. That was really fun.
Thank you, John. Thanks, everyone.